Hello friends, welcome to Bioassembling. In this session, I will discuss lots more about Golgi complex. Let's get started the session. First of all, Golgi complex. Golgi complex was discovered by Camillo Golgi during an investigation of nervous system and he named it the internal reticular apparatus. Functionally, it is also known as post office of cell. Means Golgi complex is discovered by Camillo Golgi, and when he investigate about nervous system, then he named the internal reticular apparatus. And functionally, Golgi complex is also called post office of the cell. Certain important cellular functions such as biosynthesis of polysaccharide, packaging, compartmentalizing of cellular synthetic products like protein, production of exocytotic secretory vesicle and differentiation of cellular membrane occur in Golgi complex or Golgi apparatus. Means Golgi complex functionally known as post office of the cell and its important function of biosynthesis of polysaccharide packaging, synthesis of proteins, production of exocytotic secretory vesicles and differentiation of cellular membrane. Location Golgi complex located in cytoplasm of animal and plant cell. Next we discuss about occurrence of Golgi complex. The Golgi apparatus occur in all eukaryotic cell. The exception are the prokaryotic cell, mycoplasms, bacteria and blue-green algae, and eukaryotic cell of certain fungi, sperm cell and baryophytes and pteridophytes, cell of mature sevia tubes of plant and mature sperm, and red blood cell of animal. Their number per plant cell can vary from several hundred in tissue of corn root algal rhizoids, that is more than 25,000 in algal rhizoids. Saviors in 1965 Golgi complex are present in to a single organelles or in some algae. In higher plants, Golgi apparatus are particularly common in secretory cell. Means higher plants contain Golgi apparatus in their secretory cell, and in young rapidly growing cell or young plants contain Golgi complex in their rapidly growing cell. In animal cell, they usually occur in single Golgi apparatus. However, its number may vary from animal to animal and from cell to cell. Means Golgi apparatus numbers vary from animal to animal and from cell to cell. Paraamoeba species has two Golgi apparatus and nerve cell. Liver cell and chordates oocytes have multiple Golgi apparatus. There being about 550 of them in liver cell. Means Golgi apparatus about 50 in liver cell. Next come to the structure of Golgi apparatus. First of all is flattened sacs or cisterna. Cisterna of Golgi apparatus is about 1 micrometer in diameter and flattened plate-like or secure like closed compartment which are held in parallel bundle or stalks and one above other. In each stalk, cisterna are separated by space of 20 to 30 nanometer which may contain rod-like elements or fiber. In this structure you can see the cisterna. Here these are the rod like structure and is contain many fibers, and you can see this structure. Inside the cisterna cytoplasmic fluids are present. You see the dotted structure cytoplasmic fluid. These are numbers of cisterna present in the Golgi apparatus. Each stalk of cisterna form dictosome which may contain 5 to 6 Golgi cisterna in animal cell and 20 or more cisterna in plant cell. Each cisterna is bounded by a smooth unit membrane about 7.5 nanometer thick, having a lumen vary in width from about 500 to 1000 nanometer. Means lumens contain 500 and 1000 nanometer its width polarity. The margins of each cisterna are gently curved so that entire dictosomes of Golgi apparatus take a bow-like structure. The cisterna at the convex end means the cisterna at the convex end of the dictosome comprise proximal forming or cis phase. Convex end of the cisterna is also called cis phase, and the cisterna at the concave end of the adectosome comprise the distal maturing transphase. Transphase of the Golgi apparatus are also called maturing phase. The forming or cis phase of Golgi located next to either of nucleus or a specialized portion of rough endoplasmic reticulum. Means cis phase is located in the specialized portion of rough endoplasmic reticulum. That lake-bound ribosome is called transitional endoplasmic reticulum, and trans phase of Golgi is located near the plasma membrane. Cis phase located in the specialized portion of rough endoplasmic reticulum, whereas the trans phase is located in near to the plasma membrane. The polarization is called cis-trans axis of the Golgi apparatus. Dictosome means unconnected unit called dictosome. When we talk about the structure of Golgi apparatus, you can see that these are the structure of Golgi apparatus. The The structure of Golgi apparatus is divided into four parts: cisterna, tubules, vesicle, and vacuoles. Cisterna, you can see that these are the structure is called cisterna, and tubules. You can see that the structure of tubules. And when we talk about the detail of cisterna, Golgi complex consists of a stalk of generally Golgi complex consists of a stalk of generally 4 to 8 membrane-bound vacuoles or cisterna. Unicistern dictosome are found in fungi. Means one cisterna dictosomes are found in fungi. The membrane of a cisterna are smooth but variable thickness, and it enclosed of 60 to 90 angstrom. In stalks, the adjacent cisterna are separated by a distance of 100 to 300 angstrom. Means adjacent cisterna, you can see that these are the cisterna, two cisterna, and these are separated by a distance of 100 to 300 angstrom. And the convex phase, or this two phase containing in the Golgi apparatus, convex phase or concave phase. Convex side is also called forming phase or cis phase. These are the cis phase of the Golgi apparatus. And concave side of the apparatus is known as maturing phase or trans phase. These are concave side of the apparatus, and these are convex. The forming phase or convex phase contain about 4 nanometer of thickness. It's 4 nanometer of thickness. And the forming phase or convex phase receive vesicle from endoplasmic reticulum. It received there. You can see that the vesicles come from endoplasmic reticle. These are the vesicles, blue color structure, and it's come from endoplasmic reticulum, and it received by the convex phase or cis phase of the cisterna. And let's come to tubule. The tubule form complicated network toward the periphery of maturing phase or trans phase of the cisterna, and actually tubule arise due to the fenestration of cisterna, and they have diameter of 300 to 500 30 to 50 nanometer, 30 to 50 nanometer of diameter of tubule. Let's come to the vesicle. They are small sac like, you can see that these are vesicle, and these are the small sac like of 20 to 80 nanometer diameter. Its diameter about 20 to 80 nanometer. The vesicle are found attached to the tip of tubules. You can see that these are the vesicles and it attached to tip of the tubule, and it form various level of in the network. There are the two types. Means vesicles are of two types: rough vesicles and smooth vesicle. The smooth and coated vesicle are also called rough vesicle, rough surface, and they elaborate membrane protein. Coated vesicle or rough vesicle is contain membrane protein. These are the rough vesicle and these are the smooth vesicle. And the smooth vesicle have a smooth surface and they contain secretory substance and hence the known as secretion vesicle. Means a smooth vesicle is called secretory vesicle, or because they contain secretory substance, and the rough vesicle or they contain coated protein, membrane protein. Let's come to the vacuoles. Vacuoles, these are the vacuoles. You can see that the structure is the vacuoles, or it also called Golgian vacuole. They are expanded part of cisterna, which have become modified to form vacuole. Means when cisterna is expanded, it form vacuoles. And the vacuole develop from concave or maturing phase. These are the maturing phase, and here you can see that the lots of vacuoles are developed. Golgian vacuole contain amorphous or granular substance. Inside the Golgian vacuoles, it contain amorphous or granular substance. Some of the Golgian vacuole function as lysosome, and these also function as lysosome. Some of the Golgian vacuole also function as lysosome. This all about the structure of Golgi body, and let's come to its function. These are you can see that the tubules associated vesicles of 30 to 50 nanometer diameter and surrounded to dictosome and radiated form it, and periphery area of dictosome is fenestrated or sac like structure. And I also told about the dictosome or unconnected unit is called dictosome. Let's come to vesicle. The vesicle are 60 nanometer in diameter and are of three types: transition vesicles or small vesicle, membrane vesicle, and from valve form transitional endoplasmic reticulum to migrate the converges phase of Golgi, where the coalesces of to form new cisterna. Secretory vesicle are varied in size, membrane limited, means its size varied from membrane, and vesicles which discharge from the margins of cisterna of Golgi. They often occur between the maturing phase of Golgi and plasma membrane. Means you can see that the structure secretory vesicles are often occur in the maturing phase of the Golgi body. Calythrine coated vesicle and spherical protubules about 50 micrometer in diameter and rough surface. I have also told about the calythrine coated vesicle or rough which contain rough surface, and their main function of rough surface is to elaborate membrane protein. They are found in periphery of organelle, usually at the end of single tubule, and the morphological unit quite distinct from secretory vesicle. The calythrine coated vesicle are known play to role in the intracellular traffic of membrane and secretory product. Let's come to its function. First of all, function of endoplasmic reticulum. Function of Golgi apparatus: modifying, sorting, and packaging of macromolecule of cell secretion. Means Its main function is to modify, sorting, and packaging of macromolecules for cell secretion. I am also told about that Golgi apparatus is a act like a postman. The Golgi complex is involved in transport of lipid around the cell, and the creation of lysosome protein are modified by enzyme in cisterna by glycosylation and phosphorylation by identify the single sequence of protein in question. For example, Golgi apparatus add mannose 6-phosphate labeled 2 protein distinct for lysosome and one molecule of phosphorylated in Golgi in the apolipoprotein which form molecule known as VLDL, that is constitute of blood serum, and the phosphorylation of this molecule is important to help in aid their sorting for the secretion into the blood serum. Means one molecule of phosphorylated Golgi is apolipoprotein, and this is also known as VLDL, and that constitute that form in the blood serum, and the phosphorylation of this molecule when this molecule is phosphorylated and important to help the to sorting the secretion, and it help to sorting and secretion into blood serum. Second function is proteoglycans and carbohydrate synthesis. This includes the production of glycosaminoglycan, glycosaminoglycans, GAGs, and long unbranched polysaccharides, which the Golgi then attach to the protein synthesis in the endoplasmic reticulum to form proteoglycans. Means the production of glycosamine glycans. It is a long unbranched polysaccharide which Golgi then attach to protein synthesis and in the endoplasmic reticulum and form the proteoglycans. Its third function is in animals. Golgi Golgi bodies function in animals. In animals, Golgi apparatus involved in packaging and exocytosis of the following. First of all, zymogenes of exocyrin pancreatic cell, mucus or glycoprotein, secretion of goblet cell to intestine, lactoprotein, secretion of mammary glands, and secretion of compounds called thioglobulins, thyroxine hormone by thyroid cell, and secretion of tropocollagen and collagen. Means its main function is to secretory in animals, and its main function involved in packaging and exocytosis of the following. First, zymogen in pancreatic cell, mucus in secretion of goblet cell intestine, and lactoprotein secretion by mammary gland, or secretion of compounds like thyroxine hormone by thyroid gland, and secretion of topocollagen and collagen, formation of myelin and granules, other pigments. It also help in formation of myelin granules and other pigments, and also formation of yolk and vitelline membrane for growing primary oocytes. When primary oocytes growing, it also help in the formation of vitelline membrane. It also involve in the formation of certain cellular organelles such as plasma membrane, lysosome, and spermatozoa, and cortical granules of variety of oocytes. Means in animal cell, Golgi bodies first function is the packaging of the packaging and exocytosis, and it second, it involved in the secretory vesicles, and involved in the certain cellular organs such as plasma membrane and lysosome, acrosome, spermatozoa, and cortical granules, and variety of oocytes. Fourth function is Golgi function in plant. Golgi apparatus function in plant. In plant, Golgi apparatus is mainly involved in secretion of material in primary and secondary cell wall. Means Golgi apparatus main function is secretion of granules and materials. First, formation and export of glycoprotein, lipid, pectins, monon, and cellose lignin. Means inside the cell wall, first of all, it's form glycoprotein, lipid, pectin, monomers of hemicellose and cellose and lignin. During cytokinesis, mitosis or meiosis. Cytokinesis, you know that the division of cytoplasm is also called cytokinesis. The vesicle originating from periphery of the Golgi apparatus and coalesces and formoplast area of the semi-solid and is called cell plate. Means and these vesicles, first of all, Golgi body secrete vesicle, and this vesicle originate from periphery of Golgi apparatus and coalesces and formoplast an area of to form semi-solid layer is called cell plate. This all about the structure of Golgi body and their function. Let's come to its conclusion. First of all, Golgi body is discovered by Camillo Golgi during the investigation of nervous system, and he named the internal reticular apparatus. And Golgi body function is post office of cell, and its main function is biosynthesis of polysaccharide packaging of cellular synthetic product and exocytotic and secretory vesicle. Second occurrence, Golgi body occur in all eukaryotic cell and exception in sub prokaryotic like mycoplasma, bacteria and blue-green algae, and present in eukaryotic cells like certain fungi, sperm cell, barophytes, pteridophytes, cell of mature sabia tube plants and etc. And their number vary from cell to cell. In animal cell, there are usually occur in single Golgi apparatus. However, in paramoeba, it contains two Golgi apparatus, and nerve cell, liver cell, and pteridophytes have multiple Golgi apparatus. About 50 of them in the liver cell. Third, when we come to about the morphological shape or structure of Golgi apparatus, it contains four types, and cisterna or platent. Cisterna is of two type: transphase or cis phase. Transphase is called maturing phase, and cis phase is called forming phase. And convex end of the dictosome is compression and is called cis phase. Cis phase is a convex end, and concave end is called transphase or maturing phase. The forming cis phase is located in either in the next to either nucleus or specialized endoplasmic reticulum, whereas the trans phase located in the near to plasma membrane. These are the structure of Golgi apparatus. These are the transport vesicles come from endoplasmic reticulum, and it come to the cis phase and receiving side of Golgi apparatus. Cis phase is also called receiving side of Golgi apparatus. These are the tubules. You can see that these are the structure of tubules. Is the network of when Golgi cis phase cisterna is expand to form tubules, and these are the transphase, and transphase is also called shipping side of Golgi apparatus. And when the it release secretory vesicle. Secretory vesicle are further divided into two types: rough secretory and smooth, rough or coated is also called smooth secretory vesicle. This all about the structure. Let's come to tubules. Tubules are the 30 to 50 micrometer in diameter and situated in the peripheral area of the wristlet like structure. These are vesicle. Vesicle is further divided into three types: transitional vesicle, secretory vesicle, and calithrin coated vesicle. And when we talk about function, modifying sorting and packaging of macromolecule of for cell secretion, proteoglycan and carbohydrate synthesis. Golgi apparatus in main function in the animal is packaging of and exocytosis of followings like these are the protein, mucus, etc. And in plants, Golgi apparatus in main function in plants is secretion of materials to the primary and secondary cell wall. When we talk about more about its function, its main function of to complex carbohydrate hormones, production of hormones of endocrine gland is mediated through it. Means Golgi apparatus also help in the production of many hormones. And in Golgi apparatus, second function is also function is synthesis of pigment. In cheek embryo, retinal pigment has observed to synthesis by Golgi complex. Formation of root hair. Golgi apparatus also form root hair from their mother cell and is believed to take place through the agency of Golgi apparatus. Formation of lysosome. You can see their structure. Um, Lysosome is formed from the vesicles, and this all about the structure and function of Golgi apparatus. I hope you understand well. If you like this session, please like, share and subscribe. Thank you for watching.